So, children, good morning. Yesterday we did the components of endoplasmic reticulum, which are cisternae, tubules, and vesicles. Today we are doing the difference between the two types of endoplasmic reticulum. The two types of endoplasmic reticulum are RER or rough endoplasmic reticulum and SER or smooth endoplasmic reticulum. Now, the difference between the first difference between this and this, let's write all the points of rough endoplasmic reticulum first, and then all the points of smooth endoplasmic reticulum. So this is made up of cisternae. This is made up of cisternae. You know what are cisternae? Yesterday we did cisternae. Anybody who can tell me cisternae? What are cisternae? Yes. Anybody who can tell me what are cisternae? Yes, beta. Yesterday we did cisternae. If you can tell me what are cisternae, who's going to tell me? Cisternae. What are cisternae? I have written here made up of cisternae. What are cisternae? Yes, bache? Okay, I'll unmute you. Rohan.
Good morning, ma'am.
Good morning. Now you can answer what are cisternae.
Ma'am,
Yesterday
they
we
lie
did
near the nuclear membrane, and they are unbranched structures.
the unbranched structures.
And
And what
their
else?
lumen is small,
Yes. And
and they
any other
are
point?
attached with the help of a protein known as ribofuscin.
Ribo. They are attached by help of a protein. What is the name of that? What is attached with the help of protein? What
They
is
are
attached with
attached
the
to
help
the
of
membrane,
what is
cisternae.
attached to the no. Cisternae have ribosomes on them.
Yes,
If
ma'am,
this is a
ribosomes attached
cisternae,
to the
if this is
endoplasmic
the
reticulum.
cisternae, yes, if this is the cisternae, it has ribosomes on them. This way, the ribosomes are present on cisternae, and these ribosomes on cisternae are attached by on the membrane of cisternae are attached by ribofourin. Ribofourin is a protein which attaches ribosomes on cisternae. So that was good, except for the last point where you were confused about ribofourin. Ribofourin is a protein which attaches ribosomes on cisternae. Okay. Now,
Yes, ma'am.
your answer was almost correct. So they lie. The first point is made up of cisternae, then ribosomes. Ribosomes on cisternae are attached by means of ribofourin. This is something which attaches. Now the third point is they lie closer to the nuclear membrane cisternae. And in all, we are talking about RER. So RER, rough endoplasmic reticulum, lie close to the nuclear membrane. It lies closer to the nuclear membrane. Then the next point, because in rough, why rough RER is known as rough endoplasmic reticulum? Because it is made up of cisternae, and what their surface is not smooth. They have these ribosomes on them. These ribosomes are there on them, which are attached by ribofourin, and because of this presence of ribosomes on its surface, the surface becomes rough, and we call it rough endoplasmic reticulum. So, and what is the function of these ribosomes which are present on cisternae? What is the function of these ribosomes which are present on cisternae? Yes, anybody who can answer? What is the function of ribosomes which are present on in rough endoplasmic reticulum? Protein synthesis. Yes, Sanvi, correct. protein synthesis. So what is next point? In next point we will write about the function. Fourth point helps in protein synthesis and its transport. Helps in protein synthesis and its transport. Okay. Then the next point is because they lie close to the nucleus. Yesterday I made the diagram in which I made rough endoplasmic reticulum, which is made up of cisternae. They lie close to the nucleus. So they help in transport of nuclear material also. So helps in transport of nuclear material also. Helps in transport of nuclear material also. We come to the next point, which is sixth point. Sixth point is where are these present? Where are these present? These are present in fibroblasts. Fibroblasts. Now, what are these fibroblasts? Yes. What are these fibroblasts? We have already done fibroblasts. If you remember, may I know what are these fibroblasts? Yes, beta. Yes. Fibroblasts are the special cell which forms two types of proteins. We did this in tissue, areolar tissue. Fibroblasts are the cells which synthesize special type of proteins, collagen and elastin. Collagen and elastin. So present in fibroblast cells and synthesizes two proteins, elastin and collagen. Because this is RER. RER has ribosomes. Ribosome helps in protein synthesis. So elastin and collagen are also proteins. So they help in protein synthesis. We come to the next point, seventh point. There is a protein hormone which is secreted by pancreas. Can you name the protein hormone which is secreted by beta cells of pancreas? There is a protein hormone which is secreted by beta cells of pancreas. Yes. Can you name the hormone? The protein hormone which is secreted by beta cells of pancreas. Anybody who can answer? The protein hormone secreted by beta cells of pancreas. Yes, beta. You know, you have done this chapter, hormones, chemical coordination, you have done this chapter. In this chapter, you must have studied the endocrine pancreas and the types of cells which are present in endocrine pancreas, alpha, beta, gamma cells. The alpha cell secretes something, beta cells secrete a protein hormone. Can you name it? Yes, it is insulin. Now here, yes, Rohan, you are correct, it is insulin. Now here I am very specific when I say protein hormone. Protein hormone, because this is RER, rough endoplasmic reticulum. It is rough because ribosomes are present and ribosomes help in synthesis of proteins. Okay, so that is why I am very specific when I say protein hormone helps in production of protein hormone. And what is that protein hormone? That protein hormone is insulin. So I write here beta cells of islets of Langerhans in pancreas as RER. Okay, here it helps in the synthesis of protein hormone insulin. Here it helps in the synthesis of protein hormone insulin. So beta cells of islets of Langerhans. This is one point. Then we come to the SER. Why these are known as smooth endoplasmic reticulum? These are known as smooth endoplasmic reticulum because these are made up of tubules. If you remember, we made tubules like this, which are connected to the outer, the plasma membrane. We made it like this. If you remember, we made it like this. Like this we made, and there were no ribosomes on it. There were no bead-like structures, ribosomes on it. So this is first point, is it is made up of tubules. Made up of tubules, ribosomes absent. Ribosomes are absent, so no ribofluoride, nothing like this. Now here I have written they lie close to the nuclear membrane. So an SER lies, the third point, smooth endoplasmic reticulum lie close to the plasma membrane. It lies close to the plasma membrane. We come to the fourth point. Here the fourth point says it helps in protein synthesis and its transport. It helps in protein synthesis and its transport because it has ribosomes. But in SER there are no ribosomes, so it helps in lipid synthesis and its transport. Lipids are synthesized here. Helps in lipid synthesis and can you hear me now? Am I audible now? Okay. So it helps in lipid synthesis and its transport. This was the fourth point. We come to the fifth point. It is about its location. Present in fibroblast cells, like here I have written where it is present. It forms in muscles sarcoplasmic reticulum. In muscles, it is present as sarcoplasmic reticulum. In muscles, it is present as sarcoplasmic reticulum, and here it helps in the conduction of motor impulse. What is the function here? Helps in conduction of motor impulse, and one more function is also there, and removal of lactic acid. And removal of lactic acid. So these are the two functions when it is present in the muscles. The next function of this is this helps in detoxification. Sixth function of SER is helps in detoxification. Detoxification is removal of toxic substances from the body. So it helps in detoxification. Whereas if it is present in liver, it helps in glycogenolysis. In liver cells, it helps in glycogenolysis. Glycogenolysis is, lysis is always the breakdown. Glycogen, breakdown of glycogen. That means breakdown of glycogen. That means breakdown of glycogen. Then, besides this, it is present in adipocytes. I'm writing here the 8th point. Present in adipocytes. Lipids in them. It is the fat tissue which is present under the skin and it stores fats in it. So where it helps in the synthesis of lipids. Here it helps in the synthesis of lipids. Here it helps in the synthesis of lipids. Besides this, like this, RER helps in the synthesis of protein hormones. This SER helps in the synthesis of steroid hormones. Helps in the synthesis of steroid hormone. Like steroid hormones like testosterone by the testis and estrogen by the ovaries. Estrogen and one more, progesterone by the ovaries. You can write on progesterone also. Progesterone by the ovaries. So estrogen and progesterone by the ovaries is formed by SER. Okay, and testosterone in testis is formed by SER. So both are steroid hormones. Steroid hormones, two types of hormones: protein hormones, where the precursor is the hormones which are produced are proteinaceous in nature. That means made up of amino acid. In case of steroid hormone, the precursor, that means the basic component which makes a steroid hormone, is a cholesterol. Cholesterol is fatty in nature, and the SER synthesizes lipids only. It synthesizes lipids or fats. Okay, so this is the difference between SER and RER. Is it clear to you all, children? This difference is clear to you all? Yes, beta. Anything in this you want me to repeat? Yes, bache. Okay, this is clear to you. Fine. Okay, now we come to the next topic of this. No, ma'am, it's clear. Deepika, I could not understand. You have written no, ma'am, and also you have written it's clear. I could not understand. Okay, now I'll make a diagram for you so that you understand the location of the three components of endoplasmic reticulum. I make a diagram of that. So the diagram is like this. We have a complete cell and we have the nucleus here in the center. This is the nuclear membrane. This is the nuclear membrane. This is the nuclear membrane. So we have the nucleus in the center. From the nuclear membrane arises the three components. The three components are cisternae, vesicles, and tubules. So cisternae have ribosomes and they lie near the nucleus. So we will make a small protrusion from here. It makes cisternae like this.